Hi students, we are going to start very important concept of the chapter respiration, that is Krebs cycle, or it is also known as citric acid cycle or tricarboxylic acid cycle. We already completed the process of glycolysis, glycolytic pathway we already completed. In glycolysis, glucose get converted into two molecules of pyruvic acid. But now we are going to consider only single molecule of pyruvic acid, and what happened with the single molecule of pyruvic acid we will discuss further details. One pyruvic acid get converted into acetyl coenzyme. We already studied in a link reaction, and during this process what happened? Two molecules of NADH2, two molecules of carbon dioxide, and two molecules of acetyl CoA are produced. So as we are going to consider only single molecule of pyruvic acid, so we will get only single molecule of acetyl coenzyme, single molecule of carbon dioxide, and single molecule of NADH2. Okay, so NADH2 formed during this process. NADH2 is produced here. Actually, two molecules when we are considering two pyruvic acid from NAD. Okay, and carbon dioxide. Okay, and two molecules of acetyl CoA, but we are going to considering only single molecular reactions. That acetyl CoA enzyme A combines with the oxaloacetic acid. Skeleton is already drawn by me. I am going to discuss the name of reaction, name of enzyme involved during the reaction, and cofactors if any required. Oxaloacetic acid combined with the acetyl coenzyme A, and during this reaction acetyl fraction get transfer on oxaloacetic acid. Oxaloacetic acid is a 4 carbon compound. When acetyl fraction get add on the oxaloacetic acid, it get converted into citric acid, which is a 6 carbon compound. Okay, and this reaction, name of this reaction is condensation. This is a condensation reaction. Okay, because there is a combination of two smaller molecules. One is oxaloacetic acid and other one is a acetyl coenzyme A. Okay, and enzyme required for this process is citrate synthase. This is irreversible reaction. Once oxaloacetic acid get converted into citric acid, it cannot be recovered back. Once the complete cycle takes place, then we will get oxaloacetic acid back. So oxaloacetic acid converted into citric acid in the presence of enzyme citrate synthase. Name of reaction is condensation reaction, and this reaction requires one molecule of water. So H2O get added during this reaction. H2O gets added during this reaction, and oxaloacetic acid converted into citric acid. Citric acid converted into isocitrate. This is the isomerization reaction. Citric acid converted into isocitrate. This is isomerization reaction, but this is two-step reaction. In first step, dehydration occur. In the first step, dehydration occur. Dehydration. It means that one water molecule get lost. Okay, and enzyme required for this reaction is aconitase. Enzyme aconitase is required. Aconitase functions active only in the presence of ferrous ion Fe++. Okay, and we get cis aconitate. That cis aconitate converted into isocitrate. So the complete reaction is isomerization, but it includes two things. First is dehydration, and second one is hydration. What do you mean by hydration? As here one water molecule is lost, one water molecule gets added during hydration reaction. During hydration reaction, one water molecule get added, and cis aconitrate converted into isocitrate. This required again same enzyme, aconitase and cofactor Fe++, cofactor ferrous ions. Okay, so the name of reaction and enzyme I am discussing here. Skeleton is already drawn to minimize the time required for the lecture. Isocitrate get converted into oxalosuccinate. This reaction is oxidation reaction. This reaction is oxidation reaction. So the first time oxidation occur, so we call it as a oxidation 1. Oxidation is also known as dehydrogenation. Means what? In this stage, at the conversion of isocitrate to oxalosuccinate, ऑक्सिडेशन ऑकर. ऑक्सिडेशनलाच इथं डिहायड्रोजनेशन म्हणलं जातं, कारण इथं हायड्रोजन H2 दोन हायड्रोजन ॲटम रिमूव्ह केले जातात आणि ते दोन हायड्रोजन ॲटम कोण ॲक्सेप्ट करतं? NAD ॲक्सेप्ट करतो. सो वी गेट in this stage we get NADH2 which is formed from NAD. Okay, as this is a single molecular reaction, we are going to discussing only one molecule produced of NADH2 from NAD. Why? Because isocitrate lost two hydrogen. That two hydrogen get accepted by NAD and converted into NADH2. And enzyme required for this reaction is, as there is a oxidation, I already discussed in the last lecture. When there is a oxidation or dehydrogenation, the enzyme is similar always. Take the name of substrate and write dehydrogenase at the end. So the enzyme is isocitrate dehydrogenase. Enzyme is isocitrate dehydrogenase. Okay. In oxidation reaction, dehydrogenase enzyme is required. तर कशाचं ऑक्सिडेशन चाललेलं आहे? आयसोसायट्रेटचं. म्हणून आयसोसायट्रेट डिहायड्रोजनेस एन्झाइम रिक्वायर्ड फॉर दी conversion of isocitrate into oxalosuccinate. And very important, one cofactor is required. The name of cofactor is EMN++. We see Mg++ many times, but not Mn++. And also in link reaction, when pyruvate converted into acetyl CoA, at that time also pyruvate dehydrogenase enzyme we discussed. Pyruvate dehydrogenase for the action of pyruvate dehydrogenase, Mg++ ions are required. Pyruvate dehydrogenase complex required magnesium ions. Okay, but here manganese ions are required in the conversion of isocitrate to oxalosuccinate. The next reaction, oxalosuccinate to 𝝰-ketoglutarate, is decarboxylation reaction. Decarboxylation. It means what? CO2 molecule is lost and which is removed from mitochondria. It comes into the cytoplasm. From cytoplasm to it entered into the blood. Where in RBCs is combined with the water molecule to convert CO2 plus H2O gives carbonic acid. Carbonic acid is produced inside the RBCs. Okay, that we see later on in 12th standard respiration chapter. Carbon dioxide removed from mitochondria. कारण त्याचा तिथे काही यूज नाही, एक्सक्रिटरी वेस्ट येते, त्यामुळे ते मायटोकॉन्ड्रियामधून रिमूव्ह केलं जातं. डिकार्बोक्सिलेशन रिएक्शन CO2 लॉस्ट होते. मग एन्झाइमचं नाव काय असेल? कशाचं चाललंय डिकार्बोक्सिलेशन? ऑक्झॅलोसक्सिनेटचं ना. सो द नेम ऑफ एन्झाइम इज ऑक्झॅलोसक्सिनेट डिहायड्रोजनेज. नेम ऑफ एन्झाइम इज ऑक्झॅलोसक्सिनेट dehydragenase. oxalosuccinate dehydrogenase convert oxalosuccinate into alpha ketoglutarate. Okay, so when I put the numbers to each and every reaction, we will get, this is the first reaction, condensation. Okay, this is the first reaction, condensation. Second is dehydration. Third one is hydration. Fourth one is oxidation. Fifth one is decarboxylation. Sixth one is oxidative decarboxylation. Two things happen simultaneously. Oxidation also occurs and decarboxylation also occurs. So this is the oxidative decarboxylation. Oxidative decarboxylation. It means that here carbon dioxide is also lost. Carbon dioxide is lost. And what adds up in a alpha ketoglutarate? Succinyl CoA is added. CoA is added. Coenzyme is added. So the coenzyme added, CO2 is lost, and at the same time oxidation occurs. And this oxidation is a dehydrogenation type oxidation. Dehydrogenation म्हणजे removal of hydrogen. Removal of hydrogen वाला ऑक्सिडेशन असल्यामुळे अल्फा केटोग्लुटारेटने जे हायड्रोजन लॉस केलेले ते कोण घेणार आहे? ऑफ कोर्स NAD घेतो. NAD takes up the hydrogen which is lost by alpha ketoglutarate and converted into NADH2. NADH2 is, you already know from this NADH2 in mitochondria on the inner membrane, it is through the ATS pathway. NADH2 can be converted into 3 molecules of ATP. So this is the energy. This is the energy. Check me, याला check म्हणलेले. हे काय डायरेक्ट पैसे नाहीये. हे चेक आहे, हे बँकेत टाकायचे ATS मध्ये आणि त्या बँकेतनं एका NADH2 पासून आपल्याला 3 ATP मिळतात जे डायरेक्टली बॉडीमध्ये यूज केले जाऊ शकतात ड्यूरिंग द मेटाबॉलिक रिएक्शंस, ड्यूरिंग द बॉडीली ऍक्टिव्हिटीज. Okay, so the third, so the fifth, sixth one is the sixth reaction is the oxidative decarboxylation. Sixth one is oxidative decarboxylation, in which coenzyme is added, CO2 is lost because it is decarboxylation, and due to oxidation, hydrogen is lost by alpha ketoglutarate, which is accepted by NAD, and it becomes NADH2. Arrow is important here. Arrow is important here. So one more important thing is remain to tell you, that is dehydration and hydration. These both the reactions are reversible. So we must draw, as it is reversible reaction, this aconitate can be converted into isocitrate, citrate, and isocitrate can be converted into reverse back cis aconitate. Remaining reactions, oxidation, decarboxylation, oxidative decarboxylations, are irreversible reactions. Now this succinyl CoA undergo hydration and phosphorylation. Name of reaction is hydration and phosphorylation. Hydration and phosphorylation. But during conversion of succinyl CoA to succinate, some amount of energy is lost. That energy used by guanosine diphosphate to convert it into guanosine triphosphate, GTP. जसं ATP ADP असतं तसं GDP आणि GTP असतं. GDP get converted into GTP during the energy lost by conversion, by conversion of succinyl CoA to succinate. Succinate म्हणजे succinic acid. Okay, but GTP is highly unstable, so it cannot be sustained in a mitochondrial matrix. That's why that GTP again get converted into GDP by the loss of inorganic phosphate, and that inorganic phosphate is taken up by ADP, and that ADP get converted into ATP. So we get directly adenosine triphosphate here. NADH2 need to enter into electron transport system, but इथं डायरेक्टली ATP मिळालाय ना, तर याला म्हणतात सबस्ट्रेट लेवल फॉस्फोरायलेशन. दिस इज सबस्ट्रेट लेवल फॉस्फोरायलेशन. डायरेक्टली ATP मिळाला, दॅट इज सबस्ट्रेट लेवल फॉस्फोरायलेशन. Succinyl CoA converted into succinate by process known as hydration and phosphorylation. म्हणजे इथं परत H2O मॉलिक्यूल ऍड होणार आहे. कुठे कुठे ऍड होते लक्षात ठेवा. कंडेन्सेशनला ऍड झाला H2O, म्हणजे ही पण हायड्रेशन रिएक्शन आहे एका वेळी. इथं पण H2O ऍड होते. सो दिस इज आल्सो हायड्रेशन रिएक्शन. हायड्रेशन नावाच दिलेले आहे. and this reaction takes place in the presence of enzyme succinate thiokinase. Succinate thiokinase. Succinate thiokinase. Okay, and the name of the enzyme which is involved in conversion of alpha-ketoglutarate to succinyl CoA काय असेल नाव? ऑक्सिडेशन रिएक्शन आहे, म्हणजे डिहायड्रोजनेज असणार. कोणाचं चाललंय? अल्फा-केटोग्लुटारेटचं. अल्फा-केटोग्लुटारेटचं चाललंय ना, नाव असेल याचं अल्फा-केटोग्लुटारेट डिहायड्रोजनेज. डिहायड्रोजनेज अल्फा-केटोग्लुटारेट डिहायड्रोजनेज. ओके, हे कंप्लीट लिहायचे तुम्ही, शॉर्ट फॉर्म नाही चालणार. अल्फा-केटोग्लुटारेट डिहायड्रोजनेज. नाउ this one is our seventh reaction. This one is seventh reaction. Seventh reaction in which succinyl CoA converted into succinate in the presence of enzyme succinate thiokinase. Name of reaction is hydration and phosphorylation. Okay, next. Now this reaction is reversible. This reaction is reversible, so I can show the reaction in both the ways. Succinyl CoA converted into succinate. Succinate can be converted into succinyl CoA again. Now succinate converted into fumarate. Succinate converted into fumarate. This is again oxidation reaction. So this oxidation, order of oxidation. So this is the first oxidation. This one is the second oxidation. Now this one is the third oxidation. Oxidation number 3. Okay. Oxidation is also known as dehydrogenation. Please keep in mind. Succinate च चाललंय ना ऑक्सिडेशन. ऑक्सिडेशन असतं एन्झाइम कोणता असतो? डिहायड्रोजनेज. सो succinate dehydrogenase. Name of the enzyme is succinate dehydrogenase. Okay, succinate dehydrogenase. And as there is an oxidation, which oxidation? Addition of oxygen वाला oxidation नहीं है, ये loss of hydrogen वाला oxidation है. So succinate lost hydrogen which is accepted by, here not NADH, here it is FAD, flavin adenine dinucleotide. Here it is FAD, and so we get here very important molecule, that is FADH2 from FAD. FAD पासून काय बनणार आहे? FADH2. Okay, because this hydrogen accepted by FAD from succinate. One more thing I would like to tell you. Succinyl CoA converted into succinate. Succinyl having a coenzyme. Succinate do not have coenzyme. So that coenzyme is lost here. This coenzyme is lost here. So this coenzyme is lost. And that coenzyme again entered into the previous reaction. हा जो coenzyme आहे ना, तो इथले इथेच फिरत राहतो. माइटोकॉन्ड्रियल मॅट्रिक्स मध्ये असलेला coenzyme आहे, तो इथले इथेच फिरत राहणार आहे. नवीन ज्यावेळेस पायरुवेट चं कन्वर्जन acetyl CoA मध्ये होईल, acetyl CoA oxaloacetic acid सोबत कंबाईन होईल. ज्यावेळेस परत ही रिएक्शन होती, तोच coenzyme परत अल्फा-केटोग्लुटारेट वर ऍड होणार आणि succinyl CoA बनणार. succinyl CoA टू succinate बनताना तोच CoA परत बाहेर पडणार, परत अल्फा-केटोग्लुटारेट सोबत तो ऍड होणार आहे. म्हणजे तो सेम coenzyme आहे. ओके, नाउ succinate dehydrogenase oxidation 3. fumarate get converted into malate. This reaction is again hydration reaction. Okay, this reaction is again hydration reaction. Name of reaction by blue color, name of enzyme by red color. Hydration reaction. So इथे परत आपल्याला H2 ऍड करावा लागेल. Hydrogen water is added. Okay, hydration reaction and it happens in the presence of enzyme fumarates. Fumarates. It happens in the presence of enzyme fumarates. F-U-M-A-R-A-S-E. Fumarates. Now malate number 7 reaction. This is 8th reaction. 8th reaction. 8 नंबरची जी रिएक्शन आहे ती सुद्धा रिव्हर्सिबल आहे. म्हणजे मला डबल एरो दाखवावे लागतील. ओके, एक जाणारा एरो दाखवायचा आणि एक रिटर्न येणारा एरो दाखवायचा. लाईक दिस. ओके, reaction number 9. This is also reversible, so I would like to show double arrow. This is 9th reaction. Okay, which is hydration. And now malate converted into oxaloacetic acid. This is again oxidation. Which number? Here 3. Now 4. Oxidation 4. Okay, oxidation number 4, and this takes place in the presence of enzyme. कोणाचं चाललंय ऑक्सिडेशन? मॅलेटचं चाललंय ना. ऑक्सिडेशन साठी कोणता एन्झाइम असतो? dehydrogenase. So the name of enzyme is malate dehydrogenase, and this is also reversible reaction. Name of enzyme is malate dehydrogenase. Malate dehydrogenase. So malic acid going to lost the hydrogen. Malate dehydrogenase, and that lost hydrogen get accepted by NAD, and we got NADH2. So it is very clear that in single turn of Krebs cycle, we get 3 NADH2, we get NADH2, 3 FADH2, how many? 1, and ATP directly 1 in single turn. But this going to happen 2 times for one glucose molecule. For one glucose molecule, this going to happen 2 times, because from one glucose molecule we will get 2 pyruvic acid. 2 pyruvic acid will convert into 2 acetyl-CoA. As I written all this cycle by considering single molecule, the things to happen twice when there is a single glucose molecule. From one glucose molecule, these things going to happen twice. सगळे डबल वेळ होणार आहे. म्हणजे टोटल NADH2 किती फॉर्म होतील? टोटल NADH2 from single glucose molecule in Krebs cycle there will be 6. Okay, total FADH2 produced in Krebs cycle from one glucose molecule ते 2 असतील आणि ATP पण 2 असतील. ओके, हे सगळ्यांनी लक्षात ठेवायचे. This is also known as, what is the name of the cycle? This is the Krebs cycle. Krebs cycle. Another name for Krebs cycle, because Krebs cycle or tricarboxylic acid, or citric acid cycle. Okay, these are the 3 names, because it was discovered by Dr. Hans Kreb. That's why the name Krebs cycle. Why it is called as tricarboxylic acid cycle? Because the first stable compound formed is citric acid, which contain 3 carboxylic acid group. That's why it is tricarboxylic acid cycle, TCA cycle in short. Why it is citric acid cycle? Because the first intermediate compound formed when acetyl-CoA adds on oxaloacetic acid is citric acid. That's why it is citric acid cycle. ATE जे शेवटी येतं ना केमिस्ट्री मध्ये सफिक्स ATE असेल ना तर त्याचं मीनिंग असतं ऍसिड. म्हणजे हे सायट्रिक ऍसिड आहे. इथं सिस ॲकोनिटेट आहे म्हणजे सिस ॲकोनिटिक ऍसिड आहे. आयसोसिट्रेट म्हणजे आयसोसायट्रिक ऍसिड आहे. ऑक्झॅलोसक्सिनिक ऍसिड, अल्फा कीटोग्लुटॅरिक ऍसिड, succinyl-CoA, succinic acid, fumaric acid, malic acid, oxaloacetic acid. Okay. Now I would like to explain oxaloacetic acid, which is a 4-carbon compound, citric acid, which is a 6-carbon compound, and cis aconitate will be again 6-carbon. No problem. Okay, it is a 6-carbon compound. Let's write down the carbon number. 6 carbon. Isocitrate, which is again 6 carbon. Oxalosuccinate. If there is a decarboxylation or carboxylation, then only carbon number increase or decrease. During carboxylation, carbon number increase. During decarboxylation, carbon number decrease. So you should keep in mind these things. Here 6 carbon oxalosuccinate. कार्बोक्सिलेशन डिकार्बोक्सिलेशन नाही झालेलं त्यामुळे हे पण 6 कार्बन असणार आहे. डिकार्बोक्सिलेशन आहे सो अल्फा कीटोग्लुटारेट किती कार्बन बाहेर पडला एकच आहे. सो अल्फा कीटोग्लुटारेट इज द 5 कार्बन कंपाऊंड. इथं परत ऑक्सिडेटिव्ह डिकार्बोक्सिलेशन आहे म्हणजे परत एक कार्बन कमी करायचा म्हणजे सक्सिनिल-CoA किती कार्बन कंपाऊंड आहे 4 कार्बन कंपाऊंड. नेम द 5 कार्बन कंपाऊंड फॉर्म ड्यूरिंग क्रेब सायकल असा क्वेश्चन येतो. एकच आहे फक्त ते अल्फा कीटोग्लुटारेट कधीच विसरायचं नाही. सक्सिनिल-CoA 4 कार्बन कंपाऊंड. सक्सिनेट इथं आहे का कुठे डिकार्बोक्सिलेशन म्हणजे हे पण 4 कार्बन कंपाऊंड असणार आहे. सक्सिनेट फ्युमारेट विच इज अगेन 4 कार्बन कंपाऊंड. मॅलेट किती कार्बन कंपाऊंड असतं माहिती आपल्याला मॅलेट ऑक्झॅलोएसिटिक ऍसिड आणि मॅलेट दोन्हीचा कार्बन नंबर सेम असतो C4 पाथवे. सो दिस इज 4 कार्बन कंपाऊंड, दिस इज 4 कार्बन कंपाऊंड. ओके हां तर आता या टोटल पाथवेमध्ये आपल्याला तुम्ही मला सांगायचंय की डिकार्बोक्सिलेशन किती वेळा झालेलं आहे. डिकार्बोक्सिलेशन किती वेळा झालं, ऑक्सिडेशन किती वेळा झालं. ओके फर्स्ट ऑक्सिडेशन also known as dehydrogenation. Okay, how many times it occurs? How many times? Count kara. Oxidation or dehydrogenation. One here, 2, 3 and 4. So total times 4. Tell me hydration. How many times hydration occur? And how many times decarboxylation occur? Decarboxylation occur. How many times? Oxidation occurs 4 times. Hydration वॉटर मॉलिक्यूल इथे ऍड केलेलं आहे. इथं एकदा बाहेर पडतोय एकदा आत येतोय म्हणजे एक डिहायड्रेशन एक हायड्रेशन आहे ते कन्सिडर करायचं नाही. ते नल झाले तिथं. ओके नील झालं हे कन्सिडर करू नका. ओके परत कुठे हायड्रेशन इथे हायड्रेशन आहे सेकंड अँड नाव हियर इथं. सो हायड्रेशन 3 टाइम्स अँड डिकार्बोक्सिलेशन ओनली 2 टाइम्स. इथे एकदा आहे आणि इथे एकदा आहे. सो decarboxylation 2 times in single term. Okay, now oxaloacetic acidचं जर तुम्हाला स्ट्रक्चर माहिती करून घ्यायचं असेल तर त्यासाठी अगोदर ॲसिटिक ॲसिडचं ऑक्झॅलोएसिटिक ॲसिडच्या अगोदर ऑक्झॅलिक ॲसिडचं स्ट्रक्चर माहिती करून घ्या. ऑक्झॅलिक ॲसिड इज COOH. दिस इज ऑक्झॅलिक ॲसिड. Oxalic acid. Now we want oxaloacetic acid ना. So acetic acid additionally त्याच्यामध्ये. So आपल्याला acetic acid त्याच्यामध्ये additionally add करावं लागणार आहे. मग इथे काय होतं फक्त oxalic acidचं कन्वर्जन oxaloacetic acid मध्ये करत असताना COOH सेम ठेवायचा. इथे OH लॉस होणार C double bond O and here CH2COOH. How many carbons are there? 1, 2, 3, 4. This is oxaloacetic acid, which is a 4 carbon compound in this form. This is acetic acid functional group. COH and C डबल बॉन्ड O जर हे असेल तर दिस इज ऑक्झॅलिक ऍसिड आणि जर याच्यामध्ये ऍसिटिक ऍसिड ऍडिशनल झालं तर ऑक्झॅलोएसिटिक ऍसिड. सो याचं स्ट्रक्चर जे आहे OA 4 कार्बन कंपाऊंड कसं काय तर ते असं फर्स्ट कार्बन 1 2 3 and 4. नाउ सायट्रिक ऍसिड. सायट्रिक ऍसिड इज 6 कार्बन कंपाऊंड म्हणजे त्याच्यात ऍडिशनली 2 कार्बन आपल्याला ऍडिशनली दाखवावे लागतील. तर त्याचंही स्ट्रक्चर खूप सिंपल आहे. थोडंसं जास्त म्हणजे कॉम्प्लेक्स वाटत असेल तुम्हाला, क्रिटिकल वाटते, पण व्हिडिओ स्लोली पहा म्हणजे मी काय ऍड करतोय कुठे काय लिहितोय हे एक्झॅक्टली तुम्हाला समजेल आणि ते सोपं जाईल. काळजी करू नका. ओके, नाउ इट इज अ सायट्रिक ऍसिड. सायट्रिक ऍसिडमध्ये मी आधीच सांगितलं इट कंटेन्स 3 कार्बोक्सिलिक ऍसिड फंक्शनल ग्रुप. तर आपण चेक करू 3 कार्बोक्सिलिक ऍसिड फंक्शनल ग्रुप असतात का नाही. सायट्रिक ऍसिडचं स्ट्रक्चर मी ड्रॉ करतो. इट इज C डबल बॉन्ड O COOH, here it is OH, CH2COOH, and here also CH2COOH. This is citric acid. This is citric acid, citrate. You can count 1, 2, and 3. So how many carboxylic acid functional group? COOH, RCOH, carboxylic acid functional group. So there are 3 carboxylic acid functional group. That's why the name tricarboxylic acid cycle. पहिले कंपाऊंड जे आहे त्याच्यात 3 कार्बोक्सिलिक ऍसिड फंक्शनल ग्रुप असल्यामुळे TCA सायकल पण म्हटलं जातं. आणि क्रेब सायकल का बिकॉज इट वॉज डिस्कवर्ड बाय डॉक्टर हॅन्स क्रेब. ओके, this is all about the Krebs cycle. We already discussed that when lipids are digested in our body, converted into fatty acid and glycerol, fatty acid converted into acetyl coenzyme in the process known as beta oxidation. So it is converted into acetyl coenzyme. म्हणजे प्रोटीन असतील, फॅट्स असतील किंवा कार्बोहायड्रेट असतील सगळ्यांना फायनली acetyl coenzyme मध्ये कन्वर्ट व्हावं लागतं आणि मग पुढची प्रोसेस होत असते. ओके, ह्यातले जे ऑक्झॅलोएसिटिक ऍसिड आहे किंवा ह्याच्यातले जे अल्फा कीटोग्लुटारेट आहे हे खूप इम्पॉर्टंट. हे जे इंटरमिडिएट आहेत ना हे खूप इम्पॉर्टंट आहेत. मी रेस्पिरेशनला प्लीज पे अटेन्शन, मी रेस्पिरेशनला कॅटाबॉलिक पाथवे असं म्हटलेलं सुरुवातीला. इट इज अ कॅटाबॉलिक प्रोसेस, पण रेस्पिरेशन इज अ ॲम्फिबोलिक प्रोसेस. ॲक्च्युअली इट इज अ ॲम्फिबोलिक प्रोसेस म्हणजे कॅटाबॉलिझम पण होतं आणि ॲनाबॉलिझम पण होतं ह्याच्यामध्ये. मग ॲनाबॉलिझम कसं काय होतं तर ह्याच्यातले जे ऑक्झॅलोएसिटिक ऍसिड आहे किंवा अल्फा ग्लुटारेट आहे, ऑक्झॅलोसक्सिनिक ऍसिड आहे, हे जे इंटरमिडिएट फॉर्म होतात ना, these intermediates are used to produce different amino acids. These intermediates are used to produce different amino acids like alpha ketoglutarate used to produce glutamate, okay, and oxaloacetic acid used to produce aspartate, very important, aspartate and glutamate. Alpha ketoglutarate used to produce glutamate and glutamic acid, and oxaloacetic acid used to produce aspartic acid. These are 2 important amino acids. So amino acid form झाले, असे जर खूप सारे amino acid form झाले तर ते जॉईंट होतील, प्रोटीन बनेल. म्हणजे कॉम्प्लेक्स सिंपल कंपाऊंड पासून कॉम्प्लेक्स कंपाऊंड बनतंय ना. सो इट इज अ ॲनाबॉलिक प्रोसेस आणि ब्रेकडाऊन प्रोसेस आहे सगळी पूर्ण. आपल्याला माहितीच आहे की रेस्पिरेशनमध्ये इट इज अ ब्रेकडाऊन किंवा डिजनरेशन ब्रेकडाऊन होत असतं कंपाऊंडचं, कॉम्प्लेक्स कंपाऊंडचं सिंपलमध्ये. सो इट इज अ कॅटाबॉलिक. सो कॅटाबॉलिक पण आहे आणि ॲनाबॉलिक पण आहे, म्हणून रेस्पिरेशनला ॲम्फिबोलिक पाथवे असं कन्सिडर करतात. आता लिहायला जागा नाहीये, सो तुम्ही ते मेन्शन करून घ्या. ऑक्झॅलोएसिटिक ऍसिड गेट कन्वर्टेड इनटू aspartate and alpha ketoglutarate get converted into glutamate आणि असंही लिहा that glutamate used in synthesis of chlorophyll, phycocyanin and cytochromes. Chlorophyll, phycocyanin and cytochromes. Okay, so म्हणजे काहीतरी बनवण्यासाठी यूज होतोय ना, so it is an anabolic pathway. Okay, clear. This is all about Krebs cycle. Thank you.